Welcome back to Bio 111. This is Chapter 2, Section 2. We're going to be looking at characteristics and traits. Our first part we're going to be looking at are Punnett squares. We mentioned this last section. A Punnett square is a tool that will allow us to predict the outcome of these crosses we've been talking about if we know the genotypes of the parents. All right, and so the way that a Punnett square works, you put the parental genotypes at the perimeter of the square, and inside of the square, you combine them. So if you look at this one on the left, we have a parent which is big P crossed with a parent that is little p, and in the inside, what we're doing is think of these as being columns and rows. We are looking at the names of these rows. So the top left is in row big P and column or row little p, column big P, so we get big P, little p. The next one is in a column also named big P, and in a row little p, so it's big P, little p, and we basically just repeat it for the next row. And the genotypes that you see inside these squares are the expected outcomes, the ratio, that is, the possible outcomes for the F1 generation. Now we can go back in there and cross any of these two, and we can expect something different. So we look at the F2 generation. What we're going to see is because now we have big P crossing with big P, little p crossing with big P, little p, we're going to see other traits coming out. We're actually going to see that 3-to-1 ratio because we're going to see in this first square, the top square, we're going to see big P, then we're going to see big P, little p. In the next square, the bottom left, we'll see big P, little p, and then that bottom right square, we're going to see little p coming together, right? So in this case, we're going to have one big P, two big P, little p's, and one little p. And if you remember, because big P is dominant, the three genotypes that contain the term big P in it will all have the dominant phenotype. In this case, if we're looking at purple flowers, they will all be purple flowers, and the little p will have the recessive trait, which will be white flowers. I know that's difficult to look out with the without drawing it out. I'll see if I can find you some videos to supplement this. All right, so a test cross. A test cross is used to determine whether an organism expressing a dominant trait is homozygote or a heterozygote. That is, whether they have two of the same alleles or two different. Right? Big. In this case, we're using Y's. Big Y, or big Y, little y. So if they are homozygous, the top one is what we're going to see. We're going to have they're all going to be the same, right? Now, however, if we have a heterozygous cross in here, we're going to see some variation in the offspring. All right. All right, so we hinted at this already. Punnett squares, they can predict Mendelian cross outcomes. So we're looking at this yellow. These are P colors, the actual color of the offspring seeds, the Ps. Yellow is dominant to green. And so if we cross to a monohybrid cross where we've got true breeding yellow and true breeding green. We cross them together. We're going to see this Punnett square down here. And the Punnett square predicts what we see in the monohybrid cross, which is that 3-to-1 ratio of the dominant phenotype versus the recessive phenotype, and which comes out in the F2 cross. All right, now, if you remember back to the last chapter when we were talking about the genes being split up and being recombined during meiosis and then through fertilization that happens during sexual reproduction, these genes are basically picked at random, and so there are this requires some basic knowledge of probability. All right, so probability basics. So chance events are described by probability. Probability is a given fraction or decimal equivalent over a range 0 to 1. A probability of 0 means that an event will not happen, right? And therefore is not due to chance at all. And a probability of 1 means an event will happen. It is certain and therefore not due to chance at all. In other words, we know certainly that this is not going to happen if it's 0, and we know certainly it's going to happen if it's 1. And because we know for certain they will happen, we're saying they're not due to chance, right? There's no chance that it's not going to happen, or that it will happen. So we're not looking at the 0, the 1 aspects of this. Anything that is to chance ranges between those values. All right. For example, the chance of a flip coin landing on heads is 50% or 1/2. All right. So the multiplicative law of simple probability. This is the idea that the chances of, for example, the chances of rolling exactly a 5 on a 6-sided die is 1/6. The chances, what is the chance that after you roll a 5 that you immediately roll a 4, right? For example, and what you do to get that outcome is you multiply the two probabilities. So it's 1/6. The probability is 1 out of 6 that you will get a 5 on any given roll, and it's also 1 out of 6 you get a 4, right? But what's the probability you're going to get a 5 then a 4? It's 1/6 you're going to get the 5, right? And then of those 5, of those 1/6 of all your rolls, 1/6 of those rolls will also be a 4. You'll get a 4. So you multiply them, so it's 136. So it's 1/6 of 1/6, which is 136. All right. So the overall chance of the occurrence of 2 or more independent random events in exact order is equal to the product of their individual probabilities. All right, the additive law of simple probability. All right, when there are multiple ways or tries for a chance outcome to occur, the overall probability is the sum of the individual events. All right, so an example, if one can win by rolling a 4 or a 5 with one die, then the overall chance of winning is them combined, right? So you have a 1 in 6 chance of winning with a 4. You have a 1 in 6 chance in winning by rolling a 5. Because these are 2 ways out of 6, you really have 2/6, which is 1/3, right? So they add. Right, you have 2 chances out of 6 possibilities to win. That's 1 out of 3. All right, so both laws can be combined for complex random events. The chance of rolling a 5 and a 4 in any order is the chance of a 5 first, 1/6 times the chance of a 4 second. It's 136. But if the order doesn't matter, we might also roll a 4 first, 1/6, and a 5 second, 1/6, or 136. So the overall chance is 136 plus 136 is 236, which is 1/18. All right. All right, so leaving math behind, some basic probability behind, we're going to look at another tool for um, notating genetics within a population or within a family tree. We call this a pedigree. Pedigrees use symbols, so we have a square for males and circles for females, and then what we typically do is if it has the trait, we shade in either the square or the circle. So a shaded in square is an affected male, and a shaded in circle is an affected female. Right now, sometimes we may not know the gender. Looking back, a lot of times it's used in trying to understand uh, a disease in a family tree. For instance, we might be interested in understanding hemophilia in the royal family of Europe, the Windsors that descended from Queen Victoria. And in some cases, we may not know um, the gender of an individual, particularly if they died young and it was not documented. So we indicate this with this diamond shape, this square that's been put on end. Now, if the individual has died, we just put a cross through their symbol that represents them. All right, so probability rules can be used in conjunction with pedigrees to make genetic inferences. And what we're talking about here, looking back at Queen Victoria's descendants, many of them suffered from a disease called hemophilia, which is um, basically uncontrolled bleeding. And it affected several male members of her family, descendants of hers, and looking at pedigrees, how they are affected, who was affected, who was not affected, we can get an understanding of how the genes work, looking at the probability from those pedigrees. All right, so this is a disease, an inheritable disease, alkaptonuria. It causes some various symptoms, dark urine and some discoloration of skin, some kidney problems as well. Looking at this example, this first generation, we have an affected male. Right, in this case, we have a blue is affected, an affected male mating with an unaffected female, and that cross yielded an affected male. All right, so that's that blue. When we're looking at it, it's little a, little a. All right, so we've determined that this disease is recessive. Now that individual crosses with an unaffected female, but we know that her genotype is big A, little a, and we've inferred that from their offspring. So they had an affected female, right? That's that circle with AA in it, and then two unaffected children, a male and a female. All right, so we go all the way to the left. We look at that affected female in the third generation. She crosses with an unaffected male. We do not know that male's genotype, but that crossing yields an affected female. And we go over to the far right. The third child is an unaffected female, crosses with an unaffected male, and we see that they do have a affected female, that blue circle, and then we have an unaffected male, but we don't know the genotype of that individual, because that individual could either be big A, or little a, right? Because both of those genotypes would yield the same unaffected result. All right, we know they're kind of like a puzzle. We know these phenotypes that we've inferred on here based off of looking at what we know the second generation's phenotypes are, and understanding, um, using Punnett squares and understanding what they have to be. Right? If they're unaffected, then they have to be big A, little a. If they are affected, they have to be AA. In other words, because that second generation rather, uh, there's no way for an individual in that third generation to have two big A's. Right, so the only option is for it to be big A, little a, and that's a sort of the inference that we have to make, inferences that we have to make in pedigrees to understand the way that these traits are passed on. All right, so a dihybrid cross. The parent generation, you have a cross parent that or true breeding for different phenotypes of two different traits. So dihybrid, instead of monohybrid, we're talking about two traits at a time. For example, seed shape, round or wrinkled, and seed color, yellow and green. So P generation homozygotes, YRR crossed with little y, little r, and the F1, we have big Y, big R, little r crossing with big Y, big R, little r, typical F1 cross. The only difference we're seeing here is that we now have two traits. We're looking at not just big Y, little r, or big Y, little y, or big R, little r. We're looking at both of them simultaneously. All right, what we see in the F2 is we end up with these more complex ratio, which is 9, 3, 3, and 1. All right. And we see this in a Punnett square example. We have 9 of the smooth yellow, 3 of the smooth green, 3 of the wrinkled yellow, and 1 wrinkled green. And that makes sense, right? Because the wrinkled green is the rarest because it has to have all recessive traits. If there's any of either of these two dominant traits in there, it's not going to be wrinkled and green. All right. So the Punnett square is just basically an expanded version of the Punnett square, where we're putting the parents' genes in here. All right, Mendel's second law, the law of independent segregation. So if you count for each trait individual, you'll see that each still gives a 3-to-1 ratio as in monohybrid crosses. This led Mendel to conclude that traits were inherited independently from one another. Basically, the idea is that the segregation and recombination of alleles from one trait has no effect on the segregation and recombination of alleles for another trait. Look at the mechanics of this. We're talking about the movement of chromosomes in meiosis, right? And basically, independent assortment or independent segregation when we're thinking about it in terms of chromosomes. What we're really talking about is the fact that these chromosomes, the mom and the dad chromosome, is randomly allocated to one of the two daughter cells, right? So it's not necessarily all mom goes to one side and all dad goes to the other side. They're just randomly ordered, and that's what's being talked about here. It gets a little bit more complicated because these are chromosomes, and some alleles are on the same chromosome as other alleles, and some alleles are not on the same chromosome as, or on different chromosomes than other alleles. So it gets a little bit more complicated when you begin to understand that these chromosomes are how they're structured. But this is what Mendel saw, and it's fairly insightful. All right, this is just another example of an F2 genotype and from a dihybrid cross. And it shows, and this is one thing I want you to understand about a Punnett square. A Punnett square shows all possible combinations. It predicts the ratios. It does not necessarily predict what any given single cross is going to give you, right? If you did an infinitely large number of these crosses, like flipping, like same as when you flip a coin a bunch of times, you will approach these ratios. Just like when you flip a coin one time, when you got a heads on it that one time, that means 100% of times it gave you a head, which is not what you expect, right? If you did it 1,000 times, you're going to be closer to 50-50, right, than you would be if you flipped it only 3 times. Same thing with this. All right, now, so far we've been talking about just complete dominance, right? If you've got that big Y allele, you're going to be yellow. You're going to have yellow seeds. If you have that big P allele, you're going to have purple flowers. It doesn't matter that you have that little p for the white gene. You're going to have purple flowers regardless. It's all. But that's not always the case. There is something called incomplete dominance. So, for instance, in snapdragons, if you cross a true breeding red flower with a true breeding white flower, what you're going to end up with are pink F1, right? So you're going to have well, we come up with some colors. We'll use R. So big R and little r. You're gonna all the big R, little r, little r's that you see, all 4 of them, or however many you get, they're all going to be pink. All right. If you then cross the F1 siblings, you'll get an F2 that has the following ratio of phenotypes. You're going to have 1 red, 2 pink, and 1 white. All right. So, in order to get a red snapdragon, you've got to have 2 dominant red alleles. In order to get a white, you have to have 2 dominant, or 2 white alleles. And to get pink, you just have to have a combination of the 2. All right. So in other words, the homozygous are red and white, and the heterozygous are pink. All right. So in some cases, we have multiple alleles, and they have a complex dominant structure. It's kind of like playing rock, paper, scissors, right? So in this case, we've got 3 rabbit coat phenotypes, and the C, right, the wild type, the brown fur, is dominant to chinchilla, which is this black-tipped white fur. And chinchilla is dominant to Himalayan, which we call CH. And then CH is dominant to little c, which is the albino, which is white fur. And so they have kind of this cascading dominance, and that creates a very complex pattern of phenotypes for crosses in these